Hi, in this video, I'll be discussing about ProImmune experience of evaluating immunogenicity risk in complex peptide drug products. ProImmune are the world leader in assessing immunogenicity risk using state-of-the-art proprietary technologies, and we have been in the forefront of this rapidly evolving field of generic peptide drug development. Our immunogenicity services have the fastest turnaround time in the industry, as such project can be time-sensitive. We work closely with the clients to support regulatory success, so feel free to get in touch with us to discuss in more detail. Here are some of the key challenges of developing generic peptide drugs, which give rise to either peptide or process-related impurities that can be formed during the synthesis and storage of these peptides. So what is the impact of these impurities on immunogenicity? The risk of clinical immunogenicity can be classified into innate and adaptive immunity. The innate immunity is the interaction of process-related impurities with PRRs leading to activation of innate immunity. The level of aggregation can also trigger innate immunity and inadvertent adjuvant effect, breaking tolerance. As for the adaptive immunity, peptide-related impurities may have increased affinity for MHC binding. This synthesis process may have inadvertently incorporate T-cell epitopes that can trigger helper CD4 T-cell responses, ultimately leading to the anti-drug antibody formation. We also know that the innate and adaptive immunity can also crosstalk to have synergistic immune response. First, we will discuss about innate immunogenicity assessment. Here you can find an overview of some of the FDA key expectations to be considered while designing the innate study. The assessment should include at least 3 batches of generic drug product and RLDs at near release and end of shelf life, using innate immune response modulating impurities as suitability controls tested at a range of optimized concentrations to demonstrate assay sensitivity, testing minimally manipulated drug product at the maximum concentration that does not impact cell viability, and demonstrating of assay signal recovery of cytokines, sensitivity, lowest limit of detection, and donor acceptance criteria. Typically, we will perform a 2-stage innate study where we will first do a pilot study to identify the maximum concentration that does not impact cell viability, followed by the main study to assess the batches of drug products together with the Hermes. Our ProStorm cytokine release assay uses unmanipulated and undiluted fresh whole blood, which this is the closest to a clinical setting. In order to identify the maximum concentration that does not impact cell viability, in the pilot study, we will test 1 batch of whole drug product at a titrable concentration, starting at 1:5 dilutions into whole blood. Our assay negative control is PBS and positive control is SCB. After 24 hours incubation, we then isolate the plasma and quantify this panel of cytokines using a multiplex immunoassay. Lastly, cell viability will also be measured. After identifying the optimal concentration from the pilot study, we will then use this concentration to perform the main study, typically including 30 donors assessing at least 3 batches of generic drug product and RLD. At the same time, a Hermes covering various TLRs pathway as suitability controls will be tested at a range of concentrations in the presence and absence of drug product. Our assay standard controls will be included and in quantifying our standard panel of cytokines. Here are some examples of the optional cytokines that are available, which you can request for. Lastly, cell viability will also be measured. Next, on the FDA key expectations in relation to adaptive immunogenicity assessment, they expect peptide-related impurities to be tested as they can be potentially being a T-cell epitope. The FDA has recommended assays such as the T-cell proliferation and the HLA binding assays to assess individual impurities. In particularly, focusing on impurities that are present at 0.1 to 0.5%, which are new or present in higher amounts as compared to the RLD. Adequate number of donors with a good HLA type coverage is expected as well. In ProImmune, we offer the ProMet T-cell proliferation assay to assess adaptive immunogenicity. We can test generic peptide drug product, RLD, and purify individual impurities, typically using PBMCs from at least 40 HLA type individuals representing tissue type distribution in the general population. Specific HLA distribution can be requested too. Using suitability controls such as CEFT and KLH peptide pools tested at a range of concentration to demonstrate sensitivity of the assay. Co-culture of PBMC with test articles at pre-agreed final concentration and T-cell proliferation is measured at the end of 7 days using ultra-sensitive CFSE flow cytometry assay in 6 replicates. Cell viability will be measured for all test conditions. Lastly, our review HLA competition assay allows you to test your generic peptide drug product, RRD, and purify individual impurities. This assay is ideal to confirm biological activity of HLA binding peptides and other compounds interacting with HLA. Binding affinity will be determined using 12 concentration of the competitor test articles against a known control peptide to gain a good single time constant off-rate curve. In summary, ProImmune are equipped with the right technologies to support your generic peptide project for regulatory success. Our experienced immunology specialist team is knowledgeable about the FDA expectations in this rapidly evolving field. You may find our contact details below, so feel free to contact us and our friendly staffs are happy to discuss with you in more detail. Thank you for your attention.